Rahim. I'm Dr. Sehrish Firyal, working as assistant professor in the Institute of Biochemistry and Biotechnology, University of Veterinary and Animal Sciences, Lahore. Today, I'm going to talk on one of the very important topic of the genetics, that is the use of the mitochondrial DNA for the genetic characterization of the avian species, and particularly the Pakistani pigeons. And for this current research work, we have used we have choose very important mitochondrial genes, that is the ND2 gene. ND2 gene based DNA barcode for the genetic characterization of the Pakistani pigeon. First of all, the little bit brief introduction about the avian. Pakistan is actually blessed with the very diversified avian fauna. Among them, the pigeons are one of the most important usage bird. And if we talk about their identifications, so far phenotypic methods have been used, but there are some limitations. There are some pros and cons of the phenotypic characterization and identifications of the bird. That's why we need some. There's a dire need of, and there's a need of the time for development of such barcodes, so that by using them we can characterize the birds in an authentic way. Now, this slide actually shows a little bit introductions about the pigeon, what is actually the pigeon is, and this is the taxonomical the information regarding the pigeons. Pigeons belongs to order Columbiform, and the family is Columbidae, and genus is Columba. And so far, there are 12 subspecies in the uh, Pakistan that has been characterized phenotypically on phenotypically by the birds breeders. And although by the fancier and the bird breeders, there are some traditional and there are some classical approaches for their identification, like using their body weight, their shank length, their skin color, earlobe, the ear color, neck muffs, and the feather pigmentation. All these can be contributed and can be used for the identifications, for phenotypic identifications of the birds. But there are some problems that all these methods can be affected by some other factors as well. So all these methods are the crude one. That's why geneticians and the biotechnologists, they are in a search for the development and such authentic genetics markers. By using them, we can well identify the bird species. And among them, the molecular approaches, the rapidly amplified polymorphic DNA, restriction fragment and polymorphic DNA, microsatellites analysis like VNTRs and STRs, and mitochondrial DNA analysis, they are important one. And for this current research work, we selected the mitochondrial DNA. There are some characteristics of the avian mitochondria, that there are exactly 337 genes, and this is going to span over the 15 to 17 kb approximately genome length. And this is little bit different from the nuclear DNA, and as far as its inheritance pattern is concerned, it's do not follow the classical Mendelian inheritance. The inheritance is exclusively from the maternal sides, and these genes are very important for the phylogenetic analysis as well, because there are certain regions in the mitochondrial genome which are highly conserved, and there are certain regions in the mitochondrial DNA, they are highly hypervariable. That's why it's one of the important, very useful milestones for the study of the phylogenetic studies. And there are, as it has been mentioned, that there are 37 genes, and among them ND2 mitochondrial gene is a very important, that is involved in the encoding of the enzyme NAD at ubiquinone oxidase reductase chain 2. And this is a little bit conserved region as compared to the D loop, cytochrome B, and CO1, and that's why this has been used for the identifications of the markers at a species and the subspecies and the breeds level. Phylogenetic analysis, for the phylogenetic analysis, again, this is a very important one. And under these objectives, the study has been planned. The following coordinate objectives: the genetic characterization of Pakistani pigeons through mitochondrial antidote, their identification of at their taxonomic level, and ultimately, finally, their phylogenetic analysis. And this is slide actually shows the methodology step by step by using that, by following that we have conducted this research work. First of all, the birds have been selected on the basis of phenotypic characteristics that, and then phlebotomy has been performed by the for the sake of the blood collection from the wing vein, and that is actually the brachial vein. And the DNA extraction has been done by using the organic because in the starting the blood sample was very low. And the primer has been designed for the amplification of the ND2 genes because mitochondrial full genome is available in the NCBI repository. And then PCR amplification was done, followed by the sequencing and bioinformatics analysis. This slide actually shows that how this is just we have to puncture the brachial vein, and then we can get the blood samples. And DNA extraction by using the, as already mentioned, by using the organic method, and DNA sequencing was also performed by using 3130 Excel genetic analyzers by avian prism. And following analysis has been done. Chromatogram have been studied through chromas. Homology analysis has been done by the NCPA blast. Multiple alignment of the sequences has been done by using the cluster W. And for the diversity analysis and the phylogenetic analysis, the PopGen and the MEGA 5x2 was used respectively. And this actually shows the studies part. So far, we have studied the D loop, study from B and D2, but at the moment, the focus is on the ND2 genes. These are the different areas of the Pakistan from which we have done the extensive sampling for the sake of the different breeds of the pigeons. This is the Shirazi pigeons. You can see the characteristics on the basis of which they have been selected. The lucky pigeons and the Letterox pigeons. And the sequence analysis of 22 genes actually shows that there are some SNP has been identified that can be used as the DNA barcode markers at a different position. At the 2.218 position, there is a and a G pattern change, and there at the 817 position there is a G and a change from the reference sequences. So this can be used as a molecular markers for identification. Same is at the lucky and the rock pigeons and the Shirazi and the lucky pigeons. So these are actually the SNPs and the polymorphisms which can be used for the for the development of the barcodes, and these barcodes can be well identified, can be used for the identification of their authentic identifications of the bird. So by using these marker, we can overcome the problem of the traditional approaches that can be employed for the identifications, and we can also develop the kit on the basis of these SNPs, and that can be very beneficial for the fancier and especially for the birds breeders. So thank you very much. This was for today's presentations.